Good day, this is Professor Will, CBMD PhD, and today is Tuesday, September 7th, 2021. It is 5:32 and 49 seconds, a.m. Eastern Standard Time. Today's research, cancer treatment. So let's hop to it here. Cancer treatment with built-in light. Deep photodynamic therapy. Photosensitizer irradiates itself. Date August 31st, 2021. Source Wiley. Therapies should be highly effective and as free as possible of side effects. A big challenge, particularly in the case of cancer. A Chinese research team has now developed a novel form of photodynamic tumor therapy for the treatment of deep tumors that works without external irradiation. The light source is built into the drug and is switched on selectively in the microenvironment of tumors. The classic photodynamic therapy is a non-invasive cancer treatment that is relatively gentle because of its specific spatial and temporal selectivity. And to start with a photosensitizer, they administer the region where the tumor is located is irradiated with light. The photosensitizer is excited by the light and transfers some of the absorbed energy to oxygen molecules, resulting in reactive oxygen species that destroy tumor cells. Because of the limited depth of penetration of the necessary visible or UV light in tissues, this method is only useful for shallow tumors. Researchers have now developed a new photodynamic tumor therapy that works for deep tumors. It does not need external irradiation because the photosensitizer brings its own lamp along and switches it on its own once it reaches the tumor. The intelligent drug consists of 4 components linked into a single molecule: a photosensitizer, pyrophenophore by day, a pH sensor, disopropylamina group, a polymer, polyethylene glycol, and the lamp, the amino acid tyrosine carrying a radioactive iodine, 131 isotope. At the pH values found in healthy tissues, the molecules are tightly aggregated into nanoparticles. In this compact form, the irradiation is switched off, quenched, and the drug has no effect. However, tumorous tissue has a somewhat more acidic pH value. This environment causes the pH sensor to change structure, and the nanoparticles fall apart. The irradiation is switched on, and reactive oxygen species are formed that kill the tumor cells. How does this built-in light work? As it decays, the radioactive iodine isotope 131 releases beta radiation, electrons, and gamma radiation as the electrons move their charges polarize atoms, thereby causing dipole oscillations that send out electromagnetic waves, which means light. And usually, these waves cancel one another immediately, yet in the dense aqueous environment of the cells, other than in vacuo, the electrons sent out by L131 are faster than light. In this case, the quenching of the waves is incomplete, resulting in a bluish light known as Cherenkov radiation. And this excites photosensitizers enough to destroy tumor cells in cell cultures and animal models. The new drug demonstrated powerful tumor inhibition with low toxicity and minimal side effects on healthy tissue. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.